Hello students, today we are going to discuss in detail on transfection, that is a gene transfer in the animal tissue. Gene transfer in animal tissue is called as transfection. We come across various methods in the case of the gene transfer. In the case of bacterial cell, the gene transfer methods are called as conjugation, transformation, as well as transfection and transduction. Whereas in the case of the human tissue as well as animal tissue, we call it as transfection. And let us see the purpose of transfection methods and various methods, that is including chemical methods, mechanical method, and as well as biological methods of gene transfer. What are all the advantages and disadvantages in this mechanism? Let us see in detail. What is transfection means? The transfection is the method of transferring foreign DNA or RNA and various macromolecule into the eukaryotic cell, especially by using physical methods, chemical methods, as well as by the biological method with the help of the viruses. So during the transfection, we come across various terminologies. The cells that are going to be incorporated with the DNA, foreign DNA, is called as transfectants, and stable DNA means the integrated foreign DNA is called stable DNA, and transient means which cannot able to uptake the cells which cannot unable to uptake the foreign DNA are called as transient. Here is the example for transformation transfection. In bacterial cells, they undergo the process of transformation by bacterial conjugation. In conjugation method, both male and female bacterium is called as F plus and female is called as F minus. They undergo the conjugation with the help of a pillile extract projections, whereby it is capable of transferring the plasmid DNA from one bacteria to the other bacterium, that is called as bacterial conjugation. Another method is directly the DNA is transferred through the transport proteins which are found on the surface of it, whereby the uptake of DNA is taking place, that is called as transformation. Another method is nothing but in bacteria we come across when the bacteriophage infects, directly the DNA is injected into the cellular cytoplasm, whereby the DNA uptake is taking place. So this is bacterial transduction, we call it as. Whereas in human cells as well as animal cell, the DNA is transferred to the eukaryotic cell in the metazoan condition, and in advancement of eukaryotic cells, non-cancerous cell to cell takes place by the uptake of DNA from the infection of the viruses which are capable of causing cancer, like retroviruses, especially HPV, human papilloma virus, papavirus. All these things are capable of inserting the genetic element, whereby it converts a normal cell into a cancerous cell when gets upon activation. What is the purpose and uses of transfection means? It is useful in studying the gene function and how the genes are regulated, especially the antibody production, vaccine and hormone. And it is also useful in studying the protein expression, and we can able to transfer the DNA into embryonic stem cells. And the purpose of the transfection is to useful in treating the genetic disorders, whereby we can incorporate intact gene, that is a normal functioning gene, to replace the defective gene which is present in our system, either by direct delivery with the help of viruses, as well as or by incorporating those cells into those gene into embryonic stem cells. Then later the transfer of embryonic stem cells to the system will help in treating the particular disorder. Here is the diagram which shows the direct delivery by using viruses and another one cell based delivery with the help of embryonic stem cells. Here is a flow chart which shows the transfection mechanism. The transfection mechanism is of two types, either non-viral as well as biological or viral mechanism. In non-viral we come across chemical method whereby we using calcium phosphate and protein complexes and liposome mediated transfer is taking place. And mechanical method or physical method, we call it a mechanical method by the injection, that is micro injection or particle bombardment using gene gun method, and single walled carbon nanotubes are utilized for this method. Whereas a physical methods involve electroporation, ultrasound mechanism as well as dendrimers. Bacterial vectors are used for gene transfer in the case of the plant, especially Agrobacterium tumefaciens. Already we discussed under plant gene transfer. Whereas in the case of the gene transfer in the case of bacteria with the help of plasmid DNA, Salmonella, Shigella species, Listeria monocytogenes, they are capable of causing infection in the host cell and it is lysed and the plasmid DNA is released into the cytoplasm. Such bacteria are taken and the pathogenic part of the bacteria is replaced with our gene of interest. Bacterfection. Directly the plasmid vector DNA is incorporated into the bacterial DNA complex. That bacterial DNA complex is allowed to enter into the cell by the endocytosis. Once the bacteria releases the undergo endocytosis, automatically the DNA is released into the cytoplasm of the animal cell. There are various vectors that are helpful in transferring the DNA into the animal cell. Several classes are used for the classes of the viruses are used for gene therapy. At least 7-8 such clinical trials are available whereby the foreign DNA or the DNA to be treated to replace the defective DNA or incorporate into the viral genes. This is done by ligation or homologous recombination. Thereby the defective genes are replaced in a perfect mechanism. These vectors are called as the viral vectors, in which the disease causing property is removed and the fully deleted or gutless vectors we prepare. This vector contains just 6 cis-acting elements that are required for packaging and genome replication. That is, what we do is we remove the viral vectors which are having the disease causing property, or virulence genes are removed, and we remove the genetic material also, and further the DNA of the virus which are useful for therapy in transferring our gene of interest or foreign DNA will have only the gene which are necessary for the packaging as well as for the genomic replication. So the advantage is that the high carrying capacity of the DNA will be there, and further since there is no virulence genes are present, the virus when it is incorporated into the system will not have any cytotoxic effect as well as it will not produce any disease at all. So we come across various such viruses that are used in treatment procedure for the transfer of foreign DNA. Example such viral vectors are retrovirus, adeno-associated virus, and adenovirus and synthetic vectors are utilized. Retroviruses Retroviruses are capable of packaging RNA that viruses are allowed to enter into the cytoplasm. When it reaches, the mRNA will be injected into the cytoplasm, that is translated to protein directly with the help of retroviruses. Or in the viral genome, the cDNA of the particular protein of foreign DNA is incorporated, or the gene itself directly incorporated in the retrovirus in another mechanism. When the retrovirus injects, normally the capacity of the retroviruses, what they will do is it will cut the host animal DNA and will incorporate its own DNA. It will ligate on its own to the animal DNA. So when it is going and attaching its DNA, along with it our foreign DNA also gets incorporated. Here the disease causing property of the virus is removed. The genes are removed. In that place our gene of interest is incorporated, so that directly successfully our gene of interest will be incorporated in that our system, and it will replicate along with our own DNA, and the defect of the particular cell will be eliminated. And another mechanism is adeno associated viruses, that is also capable of packaging the foreign DNA, and after viral infection, that is the virus when it infects a cell, it is incorporated into the system by endocytosis, which reaches the nucleus, whereby it delivers the DNA of gene of interest. Adenovirus is also associated. It directly binds with the receptor on the surface of the membrane, and the endosome of the virus is formed. That virusome then releases the gene of interest into the cytoplasm as well as the nucleus. Further, we mimic the property of natural viruses like retroviruses, adenovirus, and adeno-associated virus, whereby we take up only the packaging proteins and we pack artificially our gene of interest for endocytotic mechanism, that is synthetic vectors to prepared. Here is the list of various viruses that are responsible for our delivery of gene or DNA's transfer or transfection. So retrovirus has the capacity to insert 8 kilobase pairs, whereas lentivirus 9 kilobase pairs and adenovirus 8 kilobase pairs. And here the retrovirus and lentiviruses are stable in transferring, and it has random insertion sites. And adenoviruses are transient. It is highly immunogenic in nature, and adeno-associated virus is capable of carrying 5 kilobase pairs of foreign DNA into the animal tissue, and it requires an helper virus for the process of infection. And herpes simplex virus can able to carry or uptake 30 to 40 kilobase pairs of foreign DNA, and there is no gene expression during the latent infection condition by this HSP, that is herpes simplex virus. And vaccinia virus is the largest virus, which can able to 25 kilobase pairs like herpes simplex virus, and potential cytopathic effects are carried out by this virus. Adenovirus is a double-stranded genome containing virus, which can able to have an 30 kilobase pairs capacity. It is stable and high capacity for foreign DNA, and wide range of host infection it can carry out, and ability to produce higher stocks of this. Here the adenovirus that comes and binds to the receptor on the surface of a particular cell, then the virus is encapsulated by the membrane, this endosome is released into the cytoplasm. Then later the endosome is associated or bound with the lysosome that releases the or dismantles the protein coat of the virus, and the DNA of interest is released into the cytoplasm. Then later that is transferred to the nucleus. Within the nucleus the DNA is injected that can be directly incorporated into the uh, chromosome. The DNA is self-spliced by the genes which are associated with it to the our chromosome. Adeno-associated viruses are first discovered as contaminants in adenovirus associates. In association with adenovirus, adeno-associated virus is a single-stranded DNA. It's a parvovirus. It is from the parvovirus family. Naturally, it is replicating deficient in nature. It needs the help of adenovirus for its replication mechanism. So, we need a supporting virus for the replication process of this enzyme. The region of this enzyme, region of the genome of adeno-associated virus contains replication capsid protein genes, and 145 kilobase pairs of internal terminal repeats are present in it. When the foreign DNA is inserted, it replaces a cap region of this gene, and it is expressed nearer to the endogenous promoter. The new vectors are designed by removing the deletion of replication gene as well as cap genes. Rep genes and cap gene, the repetitive sequence and the cap sequences are removed, and it is responsible for the development of the new virus or recombinant virus. Here we can see that promoter sequences, cap sequence, as well as the sequence of rep. The middle region, that is which is shown in red color, is removed, and our gene of interest is added, and is added with the co-transfection, whereby the DNA is packed into the adeno-associated virus. When the virus gain entrance by endosome, the endosomal capsid, the package proteins are dismantled, and the DNA is incorporated into the nucleus, and the genome gets integrated our chromosomes. In retroviruses, normally the retroviruses contains 3 different type of genes: GAG, POL, NV genes. The POL is responsible for polymerase function, and ENV is responsible for the production of envelope protein. Normally the GAG and POL regions are taken up. The other genes are removed, that is unwanted regions are removed, untranslated regions are removed. Then the foreign DNA is incorporated into this position. Then once the retrovirus is ready to cause infection. Directly the retrovirus as LTR sequence. That LTR sequence is capable of excision, as well as it excises the DNA of the chromosome in host cell, that is animal cell. The chromosome is cut, and the LTR sequence is bound and is spliced or sealed. So it is a permanent arrangement that is made in the case of our system. The alternate approach in the synthetic mechanism, the virus-like particles are developed with papilloma viruses, L1, L2 proteins are taken up, and it is predominantly used for vaccination purpose. These virus proteins are taken up that are coated on the gene of interest. Example, the gene delivery of human polyoma JC virus, murine polyoma virus, and papilloma virus, adeno-associated based VLP viruses are taken. That these capsid proteins are stored at minus 80 degrees centigrade. They are empty capsid proteins are packed with our gene of interest, and it is allowed to infect the animal as well as human cell. Here is the mechanism, protein delivery, how it is made. The GAG protein of the VSV, how it is involved in the delivery of your gene of interest. Normally it is associated with our receptors, binding with the receptors of on the surface found on the cells, and it is responsible for delivery. Once the proteins are dismantled, our DNA of interest gain entrance into the cytoplasm. Further, we come across the use of chemicals for the delivery of DNA into the cytoplasm. Normally, the DNA is negatively charged due to the presence of phosphodiester linkage and is complexed with the positively charged chemical molecules, and it gains entrance. The first and foremost is lipoprotein complex, that is, liposomes are associated that are carrying the DNA within the inner core, and the liposomes are bilipid layers, phospholipid layers. When it is added to the animal cell, that is gene of, automatically the fusion of the bilipid layer with the liposome is taking place, thereby the DNA is directly incorporated into the cytoplasm of our cell. Another method is by using calcium phosphate. The calcium phosphate is precipitated on the surface of the cells, that allows or changes the permeability of the cells, thereby the DNA can gain entrance into the nucleus by the coated calcium complex. But here 1 to 2% efficiency is taking place, that is, by phagocytotic mechanism the DNA gain entrance. So only 1 to 2% transfection efficiency is there. Another method is transfection with polyplexes or using a polycationic complex. Normally synthetic polylysins, polyamines, polyethylamines, and dendrimers are utilized for this purpose. They adopt the method of osmotic shock with a treatment with chloroquine, whereby the DNA gain entrance into the animal cell. Here we can see that polymers are polymeric nanoparticles with the DNA. The nanoparticles uses electrostatic interaction on the surface of the cell membrane, then they enters by endocytosis, then they fuses with the lysosome, thereby the gene of interest is delivered into the cytoplasm, that gain entrance through the nucleopore and gets attached to the nucleus containing chromosome. The transfection with liposomes and lipoplexes, normally we call it as lipofaction. The 90% of the cells are transfected with the help of our lipofaction method. Here we can see the diagram of lipofaction. that is lipoplexus, that is DNA with liposome is called as lipoplex, that directly moves by endocytotic process, and within the cytoplasm the lysosome fusion, the delivery of DNA takes place, and the DNA gain entrance into the nucleus. Mechanical transfection, or in which the microinjection, particle bombardment and a single walled carbon nanotubes are utilized in animal cell like plant cell. In direct microinjection, using a fine needle, the DNA is transferred to the embryonic stem cells. The direct transfer of DNA takes place in which by holding the embryonic stem cells as well as eggs and oocytes, by holding to the tip of the micropipette, then the DNA is injected into the tissue. The principle is utilized in vitro fertilization technique for the infusion of a sperm into the nucleus of the ovum. And bioelastic particle delivery, in which with high velocity the delivery of nucleic acids are taking place by coating the gene of interest over the bioelastic particles, that is tungsten as well as gold particles are coated with our gene of interest, and it is added. Normally the forceful blast of the bioelastic particles takes place with the help of the argon gas, that is present, argon or helium gas in the piston, so that it is incorporated into the plant cell. The same manner, this method is used to transfer the DNA into the skeletal cells of the animal cell. This is the latest technology in which the carbon nanotubes are utilized for transfer of genetic material. Graphene single-walled nanotubes and multi-walled nanotubes are acting as a carrier of gene of interest. Normally the diameter of these will be around 1 to 5 nanometer, length will be 550 to 200 nanometer. It is successfully involved in the delivery of siRNA delivery. Here we can see that how they are involved in the transfer. And physical methods include electroporation, ultrasound and dendrimer method. Already we discussed how the electroporation method, in which pulses of electricity is applied to the cell membrane, thereby it alters the permeability for the uptake of gene to the in vivo. This method is utilized in incorporation of DNA into skin, muscle and certain tumors, as well as internal tissues such as liver for this purpose. Ultrasound, here alternately the ultrasound waves are applied to the surface of the cells. That alternate application of this ultrasound waves is responsible for the delivery of the DNA into the in vivo of the particular cell. Another one is polyamino domain. Dendrimers are utilized as the physical method. These polycationic cascades binds with our DNA of interest, and they are undergoing transfection by addition to the cell surface. Once it is uptaken by endocytosis, the dendrimers are dismantled and the delivery of the gene that takes place to the cytoplasm. Further, it is moving to the nucleus and gets attached to the chromosome. This dendrimers Use of dentimers is a physical mechanism where we make use of it. What are all the advantages and disadvantages? The ability of the DNA which is transferred to the cells as well as by the membrane with the help of liposome mediated transfer is highly efficient when compared to the other methods. On long term study it is not having any side effects. The disadvantages in the case of the chemicals, the reagents are not useful for longer term studies, and transfection efficiency is depending upon the cell health, quality of the cell, quality of the DNA what we are using, and direct microinjection biolistic methods are highly expensive and it requires highly trained officials for the incorporation. So far we discussed various methods of incorporation or DNA transfer or transfection for the successful delivery of DNA into the tissue culture as well as into the cells. Thank you.